All right, let's start. So, what I have given you guys on the green piece of paper comes from No Brain Too Small. Really, really great resource for anything science related. They do biology, chemistry, physics as well. What I have grabbed from them is a list of all the things that we need to cover for the genetic variation external. They also do one for the other externals as well. So if you're planning to pick up the gene expression one, I'm happy to print you off the copy of that as well, so you can use it as a checklist of things that you're trying to accomplish. What I want to do first off is just have a look at the list to see what we've already covered and if there's things you want me to revise. The other thing that is on that green piece of paper on the back is a list of the key vocabulary words. This is really important because biology is very literacy based, and you need to be able to know what words to use when, and you also need to be able to read and decode the question. So it's really important that you make sure you know these terminologies. A lot of the achieved points are just writing down definitions, so it's worthwhile just going through and making flashcards so you know what these different words mean. So again, it's just like our own kind of class personal audit to see where we are and what we still need to cover. Sound good? All right. So let's have a look at some of the things we have talked about. So I'm just grabbing a highlighter so we can highlight things that we've covered but might need to revise. All right, and let me just maybe make that a little bit brighter so you guys can see it. Okay, so genetic variation and change involves the concepts of sources of variation within the gene pool and factors that can cause change in a allele frequency in the gene pool. We did a little bit on the sources of variation because we talked about mutation, but I wouldn't say we finished that topic, but I will highlight it as something that we have gotten some information on. All right, second one is biological ideas, processes relating to sources of variation within the gene pool selected from, and there's a whole list of things that they are looking at. First one is mutation as a new source of alleles. We have covered that. We've talked about independent assortment, segregation, and crossing over during meiosis, so I'm highlighting this one as well. Oh, careful. We've also talked about monohybrid crosses to show co-dominance, incomplete dominance, lethal alleles, and multiple alleles. Fourth bullet point is dihybrid inheritance with complete dominance. I know we definitely have not covered that. The effects of crossing over and linked genes in a dihybrid inheritance, we have not covered that. And then the last one, I know we definitely haven't covered any of them. It's biological ideas and processes relating to factors such as allele frequencies in the gene pool. And the three choices are natural selection, migration, and genetic drift. I know we have not covered that. So that's kind of giving you an idea of where we currently are. And I want to ask you if there's stuff that we need to revise for this lesson. And we'll just go through the list. This one I know we're going to have to revise because I'm going to integrate more material in there. So let's look at this first bullet point here about mutation and the source of new alleles. First off, do you guys remember what mutations are? Change. Do you want me to write down some definitions? You can look at this list and we can tick off mutation. So mutation is a change in the sequence of DNA.
When do we write that?
So you can write this on your notebook. You can type it up on a document. It's up to you. And I will scan everything that I'm doing so it will be available online as well. So if you miss something, it'll be there. Mutation is the change in the DNA sequence. So for example, if I have a sequence, we know that there's A, T, G, and C as our options. And I'm just going to write a random code. You guys remember we did those DNA bracelet activities? Yeah. So that's my code. And if I have a mutation, one of those things could change. Something in that sequence changes. So maybe I just have one of them change into a G over here. So this T has become a G. That's a mutation. Does this ring a bell? All right. So mutations. Do you guys remember the term allele? What's the difference between a gene and an allele? Oh, Mrs. Stumped ya. This is why we're doing this, okay?
Isn't allele the actual, wait, is it the letters?
The letters, yes. Yes, it's a good way to describe them. All right, so we have our DNA sequence. I'm just drawing a strand of DNA. Really, really poorly. Oh my goodness, that's so bad. That's what happens. Okay, so you have your helix of DNA. You're gonna be kicking yourself because you're gonna remember everything the moment I describe it. All right, so that's my DNA, right? If we have a section of DNA, what do I call that? Gene or allele?
Gene.
Good. So a section of DNA is known as my gene. So my gene is my trait. Say, for example, eye color. Yeah? And then that sequence can be different. So these are examples of two different sequences. Yeah? Same gene, different sequence. Different.
So you're gonna scan this?
I will scan it. And I'm recording this as well, so if you want, you can watch it again. Different sequence. So you see how these two have different sequences? We have that one mutation. So that is your allele. So say, for example, this allele codes for brown eyes, and that one codes for blue eyes. And so when we're thinking about that mutation is a source of new alleles, mutations are a source of new alleles because we can, that mutation can create a new trait. So that's where all these different variations come from. But we're basically 99.9% identical. So there's not that many mutations that make us different, but it's amazing. And that makes sense because we need to have the same bodily functions, right? We need to be able to digest food and breathe and do all the insulin stuff. So are we okay with those terminologies? All right. And one of the other things we talked about with alleles is dominant and recessive.
Oh,
Yeah? So you can have dominant, and then you have recessive. So say, for example, if we're talking about eye color, capital I or capital E, lowercase e is how we usually communicated them across. This ring a bell?
Yeah.
Yeah? And then you had things like, so these are my alleles. The two alleles make up the genotype. So you had capital E, lowercase e, lowercase e, lowercase e. You got two alleles, one from each parent. That one's homozygous, dominant, heterozygous, homozygous, recessive. Do you want me to write those down or you remember them? Write it down. It's a crash course in what we've been doing. And you guys remember the easy way to remember this? Homozygous, heterozygous. It fits the same way like when you think homosexual, heterosexual. And with the homozygous recessive allele, that trait only gets expressed if the dominant one is absent. So if I had two capital E's, I would expect brown eyes, because we're assuming brown is dominant. If I had a capital and lowercase, I'd have brown eyes. If I had two lower cases, I would expect to have blue eyes. And that's how things can skip a generation. All right, are we okay with that so far? Yeah? Okay, so on our list, we can tick off, we might actually need to take a little bit more time with mutation, because there's different types of mutation, if you guys remember. Do you guys remember the different types of mutation that you can get? There was, I'm trying to remember off the top of my head, point mutation. So point mutation is when you get one swapped over. So that one's a point one. Yeah. So you switch one for another. There's also silent mutations, so mutations that don't affect the outcome of the trait. There's also frame shift. Do you guys remember what frame shift ones are? So frame shift is when you add or remove a nucleotide. So for example, if I had the fat cat sat on the mat, that's my sentence, right? If I, thinking about my cat, if I delete a letter, say for example, I delete this A over here, I shift the reading frame, because remember when we were talking about reading the nucleotides, we read them in sets of 3. So I would have the fat, you see how that makes no more sense?
Oh, yeah.
Frame shift. Everything after that makes no sense.
The fat cook.
And then same thing would happen if I added an extra letter. It would just shift down everything. Yeah? Whereas a point mutation would be, trying to think of it. Get another color. I'm just mindful of the time. Trying to think of another word. Fat, mat, hat. Oh, the hat cat. There we go. The hat cat
Mat.
sat on
Mat.
the mat. I should have probably not done that in pink.
Black.
All right, I should have probably kept that in blue. Never mind. So you see how in this case I have a point mutation? I've changed the F to an H. The sentence overall still is not affected, but it doesn't make as much sense. So that is an example of a point mutation. You just changed one of the letters. Yeah, Jessica. The bell's gonna go in a minute. Yeah. Class is done. All right, so let's tick off some stuff. I think we're okay with this one here. Mutation is a source of new alleles. And then looking at the words that we've covered so far on the other side, we've done allele. We've done...
Do we keep these?
Yeah, you can keep those. Oh, I do want the audit sheet back. Yeah, because I want to just have a read over it. I'll collect this up, or sorry, I'll tick off the rest of them later. But that's what we're doing today. So just to recap, oh, I don't have anything there. You guys should have a plan of attack. I do want to collect your audit sheets, so that way I can write some notes on there about what I recommend as well. And then you'll keep going. People with post-it notes, you're not leaving until I can double check you've gotten your work done. When did you get done?
History.
You work on history. All right, what did you get done in history? Okay. I want more specific goals next time, okay? Well, let's get done. Read everyone's on it, and people with post-it notes. I want your post-it notes. And I ask you what it was. Finish the first source. Did you do that? Is it all done? So, okay. Did you do yours on paper or online? Okay, so I can read it and do that. Okay, cool. What was the goal? What were you pushing? Okay, next time, more specific. What are you working on? What are you going to get done in that paper? Huh? Alright, so you see how Savannah has a more specific goal. She's working on the same thing as you. She said, I want to finish my first source. So that's what I'm looking for. Alright. Draw pair of students, start right. Yep. And so cool. So yeah, the thing to think about is what's realistic. I don't want you to bite off more than you can chew and then get overwhelmed and anxious. So realistic goals. Realistic goals. We can't get everything done.